It's my distinct pleasure to introduce the next speaker, Georg Petzold, who also happens to be my dear friend and former colleague. Georg received his PhD from the IMP in Vienna, Austria, studying E3 ligase biology and cell cycle regulation in the lab of Jan Michael Peters. And in 2014, he joined the lab of Nico Thome at the FMI in Basel, where he did really seminal work in the TPD field, which elucidated the structural basis of how thalidomide analogs work, and he described the ever so important G-loop as the structural diagram that emits leverage. And in 2021, Georg joined the team at Monterosa Therapeutics in Basel, aiming to really unleash the full power of this compound class. And today he'll tell us about how at Monterosa the team has been able to teach CRBN some new tricks. So Georg, the floor is yours.
Thank you, Susanna. Thank you for the very kind introduction. Let me share my slides real quick. You can see my screen, right?
Yes.
Perfect. Thank
Not
you.
the slide. Okay, now it works. Perfect.
Thank you, Susanna. Thank you for this really flattering introduction. And also, again, thanks for the invitation and the chance to actually present our data here in this really cool forum. I'm really impressed how all this developed. Is clearly one of the more pleasant leftovers of these rather unpleasant Corona times. And I'm really grateful to everyone who keeps it alive since then. So you're really doing a great job, managing the webinar series, and it's really a fantastic resource for the field as well. And so, so the topic of my talk will be teaching CRBN new tricks, which essentially alludes to the existence of new binding modes on CRBN. Can you see my, sorry, I just have to clean up my screen a little bit. Sorry. Okay, super. Yeah, so it's teaching CRBN new tricks, which alludes to the existence of new binding modes on CRBN. But before I actually get to the science, I would like to spend a couple of words talking about Monterosa, who we are and what we do. And so, as many of you know, Monterosa Therapeutics is a clinical stage company, and our mission is to unleash the full potential of molecular glue degraders. And in order to achieve this, we are building CRBN-based MGD libraries with the aim to unravel the full extent of the canonical diagram, and I will explain what I mean by that in a minute, but also to explore options to actually move beyond the canonical diagram space, meaning to identify new possible ways for targets to actually engage the CRBN substrate receptor. And so the idea was that once the first two goals have been achieved, that we are extracting all the possible learnings in order to apply them also to other E3 ligase substrate receptors to eventually also move beyond cereblon. Now, in order to read the MGD libraries that we are building, we established our so-called Queen engine, which is our platform for the quantitative and engineered elimination of neo-substrates. And for this, we are developing a number of proprietary tools, such as AI and machine learning approaches, which now, for example, can assist in silico diagram prediction, but they are also helping with like ternary complex modeling. We have a strong focus on structure-based approaches to investigate ternary complex formation, apparently. And these approaches then also inform our computational methods and also feed back into them in order to update our algorithms. We also established a specialized suite of proximity screens, some of which are also developed internally to build assays with increased sensitivity to really allow this detection of these early weak events during ternary complex formation, which is really so important in the field to make early discoveries, as I'm sure many of you are aware. And we are also having strong internal capabilities for target agnostic readouts using state-of-the-art proteomics approaches. So as I told you before, the initial focus of Monterosa was to explore the full extent of the canonical diagram space, meaning to expand our knowledge around known CRBN diagrams. So as many of you know, known CRBN neo-substrates share a common structural motif, and this motif is called the G-loop. The G-loop is a relatively simple and generic structural motif that is conserved in shape, but not in sequence. In fact, the only sequence commonality between G-loops across different targets is a glycine residue that sits in a defined position of this loop, which is why it's called a G-loop. And so G-loops can be found in different domain types. And if these proteins are now overall compatible with the CRBN surface, G-loops is everything you need in order to get CRBN recruitment and target degradation. And because G loops are so generic and don't have a strong sequence preference, you can now use geometric descriptors of that loop to mine the human proteome for the existence of other G loop targets. And if we do this, we identify over 2,500 proteins with high confidence that we would predict now to contain potential G loop-like motifs. And this type of analysis now kind of defines also the target space that we are dealing with, which is, in my opinion, quite a sizable space. Now, in this analysis, we are identifying, of course, domains that have been that are known to carry G loops, like zinc finger domains and kinases, for example. But a large majority of the domains that are shown here now haven't been linked with this modality yet. In many of these cases, the domains would be considered undruggable under normal circumstances, which was, of course, really exciting to see for us. And so what this analysis also can do is now to essentially create a so-called Degron catalog, if you will, and then we can use this catalog to intersect it with our experimental data coming from proteomics, for example, which can now help identifying hits for our modality, for our approach, because a predicted G loop targets would light up in our data. And so once a new target has been identified, we can then move to nanobread, for example, for orthogonal validation. But we can then also use this technology to actually probe the involvement of the predicted G loop. And what I'm showing you here is now 5 new domain types that haven't been linked with this modality before. And as you can see in our nanobread experiments, we are picking up a signal indicating ternary complex formation. And importantly, this signal is then eliminated by the well-known G2N mutation, like the G loop mutation, kind of confirming that the recruitment of these different domain types actually happens through the G loop. So all these analyses that I've shown you so far, of course, highlight the blessings of such a generic motif, which is that it allows for a pretty large target space. But there's a flip side to this as well. So this is, again, the volcano plot that I showed you before. And as I explained already, it is intersected with our Degron catalog. And so the dark blue dots now indicate proteins that we would predict to contain a G loop. So this is a proximity ligation experiment. So it's recruitment-based, meaning that these proteins, if they get in proximity to CRBN, they are biotinylated. Then we can enrich for them in IPs compared to DMSO control samples. So we are then essentially interested in this upper right quadrant over here, which would now indicate proteins that have been proximal to CRBN. And so what stands out pretty clearly is that in this one experiment, with just one MGD, we are easily enriching for 8 to 10 different G loop targets. And so some of them, as you can see, are known CRBN neo-substrates, but every once in a while, we are also discovering new ones like NEX7 in this particular case. Right? So it's obvious from this experiment that the compound that we are using here is probably not the most selective one, but it highlights very nicely one of the key limitations of such a generic motif, namely that selectivity may become an issue. So whereas the target opportunity is really on the bright side of things when it comes to such a generic motif, the real question becomes whether G-loop-directed MGDs can ever be selective. So we at Monterosa think that they can be selective, and we in fact have like a number of internal examples where we managed to engineer selective MGDs against different G-loop targets. And there are also a number of examples in the literature and also at conferences that show that it's possible. And for the sake of this presentation, I would now like to use NEX7 as an example to show how selectivity could be achieved in this particular case. So just some background on NEX7 real quick. So NEX7 is a kinase that licenses inflammatory signaling by facilitating NLRP3 inflammasome assembly. And it does this in a kinase-independent manner. Meaning that NEX7 fulfills a scaffolding function in the assembly process, which highlights very nicely why a degrader approach in this particular context is very suitable. And so the therapeutic hypothesis here now would be that NEX7 degradation would silence this entire pathway and therefore could alleviate symptoms that arise from NLRP3 overactivation or mutation that frequently occurs in metabolic disorders like gout, for example, but it's also observed in cardiovascular diseases. So now, if you look at the G loop of NEX7 and compare it to one of the liabilities in the experiment on the left, like CK1 alpha, both being kinases apparently, so it's a very valid comparison here. So if you compare the two G loops, you will see that they are literally identical in shape. And we also observed a high degree of surface similarity to that of CK1 alpha, which again highlights the fact that achieving selectivity here could be a major challenge. However, when we determined the structure of the NEX7 ternary complex, we observed crucial differences to that of CK1 alpha, namely that the poses of the two targets on CRBN were slightly different. And this is now shown on this slide over here. So on the left-hand side, you can see a close-up of the ternary complex, which again confirmed the involvement of the gene loop in NEX7 recruitment, which is now shown in yellow over here. And on the right-hand side, I tried to emphasize now the different poses of the two targets by this animation, where NEX7 now takes the most extreme pose in this animation, the most differentiated one from that of CK1 alpha. So I hope from this animation, it's obvious that the NEX7 G loop is slightly offset. And as you can imagine, this of course now changes the interface between the backside of the G loop here and the NEX7 MGD, which now provided us with an opportunity to develop the chemistry and dial in selectivity. So this was really exciting for us to see, but it was not the only difference that we observed. So what we also saw in the ternary structure is that NEX7 actually forms a slightly larger PPI interface with CRBN, because there was now also this N-terminal portion here that contributed to the binding, which is now shown in this space-filling representation over here. Right, so this, and as you can see here, the N-terminal portion was now really binding relatively closely to the G loop and the NEX7 MGD as well. So it is possible that this N-terminal portion of NEX7 is responsible for the G loop offset. But what was also interesting to see is that this N-terminal portion actually inserts in the cleft between the N and the C terminal domain of CRBN. And by doing that, it seemed to stabilize CRBN in a partially open conformation. So this was another really exciting observation from this ternary complex structure, because it suggests that potentially a larger spectrum of CRBN conformations might be potentially accessible for neo-substrate engagement, and that CRBN is potentially more versatile in actually accommodating different target shapes, which wasn't necessarily appreciated before, at least not to such an extent. So as I mentioned before, these structural insights now helped us to rationally develop the chemistry for NEX7. And this is now the profile of our DC candidate for NEX7 that was just disclosed earlier this month, MRT 8102, which, as you can see now, is a very potent NEX7 degrader. It has a great selectivity profile over known CRBN neosubstrates, as you can see in the Western blot over here, also to some of the earlier liabilities that we saw in the volcano plot, for example, like CK1 alpha is completely untouched by this compound now. But it's also extremely clean in global proteomics. We have great ADME properties of this compound. It's orally bioavailable, and importantly, it shows positive and prolonged pathway modulation in a number of PD models, including non-human primates as well. So I think the NEX7 is really a great example to illustrate how selective MGDs can be developed for rather challenging G-loop targets, which in my opinion is really excellent news for all the great opportunities that really exist in this large target space. So now, shifting gears a little bit and moving more towards the non-canonical target space. So what we realized early on is that particularly in target agnostic readouts, is that every now and then non-G-loop targets could be proximal to CRBN. And there's, of course, a number of explanations why this could be, the most obvious one being complex formation of a non-G-loop target with a bona fide G-loop target, and then indirect recruitment through that G-loop target to CRBN, which is something that we can observe for interaction partners of CK1 alpha, for example, where we frequently observe the FAM83 proteins also in our experiments. But even if you would apply now all the possible filters to actually eliminate this bystander recruitment, there was oftentimes convincing recruitment evidence left on the table, which clearly made non-G-loop recruitment undeniable. And what's really important here to mention is that we do not only see recruitment of non-G loop targets to CRBN, but this recruitment also translates into degradation. As you can see here, the target protein that's indicated is the exact same protein, and we also see degradation for this one. So we are clearly talking about productive ternary complex formation here, which was really exciting to see. So the target protein that I'm showing you here is actually a kinase, and kinases were the first protein class where we actually saw this phenomenon, at least in our hands. But we now also have plenty of evidence for new binding modes across multiple different domain types and also different target classes. And I will come to these a bit later. So this is now another experiment here that I'm showing you. This is, again, a degradation experiment where we are using a promiscuous kinase degrader, which down-modulates a number of different kinase proteins. Most importantly now, the top scoring ones, again, are lacking any G-loop prediction. It's important to mention here that this is not the promiscuous kinase protag. It's an MGD, and we also know by now from a larger body of structural evidence that this MGD does not act through the active site of these kinases. It's mostly surface patches on the kinase domain itself or on associated domains that otherwise would be considered undruggable. Now, I'm also trying to illustrate this here now. So the first three examples that you can see are all kinase domains on CRBN. The purple one is the G-loop post of CK1 alpha as a reference. And I hope you can appreciate that in the other two examples, the pose of the kinase domain is completely different. And then the last one is actually a associated domain to a kinase. And now if we take a closer look at the compound interface, I hope it's clear to everybody that these novel binding modes are clearly differentiated to the canonical G-loop binding, and that there's no beta hairpin loop involved in their recruitment anymore. There are not even beta sheets involved. It's only helices here. And also the three different novel binding modes that I'm showing you here are clearly differentiated to each other. So from these examples, we were kind of thinking that it might be possible that we are maybe dealing less with structurally defined degrons, but maybe rather with surfaces that are highly complementary to CRBN, where now the MGD obviously plays a crucial role in shaping the interface properties. So if this was true, we thought instead of using geometric descriptors now to mine structurally defined degrons, we thought maybe it would actually be possible to determine so-called surface fingerprints that, for example, could be extracted from the many ternary complex structures that we have solved before. And for this, we developed some computational approaches. One of them is called phasic mimicry, which now can be used to identify similarly looking surfaces on candidate proteins, meaning that we can potentially project now surfaces from known CRBN targets to candidate proteins within the human proteome. And the one example that I wanted to show you in this context is the VAV1 target. So VAV1 was identified in one of our proteomics experiments, which you can see here. And although the initial data that we had on VAV1 was rather weak, it suggested that VAV1 could be accessible to our approach. What was interesting about VAV1 is that it didn't have any G-loop prediction, but when we used now surface mimicry, we could detect actually a strikingly similar surface on VAV1 that was kind of mimicking the surface on GSPT1 that was close to the GSPT1 G-loop. So I'm mostly talking about this concave surface over here that is sitting right next to the G-loop of GSPT1 in this region, which is now kind of mimicked by the VAV1 surface that I'm showing you on the left. And so there was some surface resemblance, but there were also physchem properties that were similar. So the surfaces were similar, not identical, but very clearly similar. So although and what's really nice about this example is that although the initial evidence for VAV1 that we had was rather weak, combination of data that I'm showing you right now could actually give you confidence that we might be dealing with a progressible target here. So VAV1 is a protein that was identified as a key mediator of B and T cell receptor signaling in a genetic screen. And we think a VAV1-directed MGD could have broad utility in autoimmune disorders. So there were many good reasons for us to really develop the chemistry here. And when we then finally determined the ternary structure of VAV1, we found now similarities, but also differences to the GSPT1 diagram. So on the left-hand side, you can see GSPT1, which is known to be a bona fide G-loop target, where the G-loop, forms interactions with more proximal parts of the MGD closer to the trip cage down here. And these interactions with the MGD and CRBN are all backbone driven. As I hope you can appreciate, the side chains of this loop now would basically point away from the MGD. But we also knew from that structure now that the rest of the protein kind of arches over the compound binding site on CRBN, and then also makes extensive interactions with like more distal parts of the MGD up here. And this arching over element now was also resembled in the VAV1 diagram, although the underlying ternary structure now was completely different, which was really exciting to see. What we also saw in the ternary complex is that VAV1 now uses this loop-like structure here for making contacts with like more proximal parts of the MGD down to the closer to the trip cage down here. But now this interaction was more sidechain driven. As I hope you can see that the amino acid side chains are now actually projecting towards the MGD in this particular case. So there was clearly a sequence component now in the recruitment of VAV1 that is clearly absent when we are talking about canonical G loops. So with everything I just told you about the VAV1 binding mode, it is clear that we are also dealing with a novel binding mode here. Hence, we called it novel binding mode number 4. So this is now the profile of our DC candidate for VAV1, MRT 6160, which was disclosed in May last year. And again, I hope you can appreciate that we are seeing potent VAV1 degradation both in B and T cells. It has an outstanding selectivity profile, which can be appreciated in Western blot and global proteomics. We have extraordinary ADME properties. It's orally bioavailable. And importantly, we see positive responses in a variety of preclinical PD models. So we are really excited about the broad utility of this molecule in the application of autoimmune diseases. So everything I told you now also about the VAV1 diagram, I think is clear that it can be added to the panel of novel binding modes now. And I think it makes sense to pause for a second here and think about some of the implications of all this. So I think it's obvious that with these novel binding modes, we clearly have an opportunity to broaden the target space, even beyond the canonical diagram space that I talked about before, right? So one can now, of course, try to generalize these, what we are doing right now, and you can approach this both from a structural perspective, but also from a surface perspective. So the second implication of alternative binding mode, which is really exciting in my opinion, is that it could, of course, offer an opportunity for MGD selectivity, where you can easily imagine a scenario where like a compound modification would dial out one binding mode, but leaves another one intact. And that's a really powerful, a really cool possibility that we have there. But I think the most important and probably most exciting implication, in my opinion, here would be that we don't necessarily need a Degron hypothesis in order to screen a target, because we can only speculate about what else could be out there. And so what we did was we actually started to screen high-value targets at risk. And what we are finding there is really spectacular, in my opinion. So I just wanted to share one last example. Clearly my favorite example so far, because it has major implications when it comes to considerations like druggability, pre-existing druggable pocket, drugging the undruggable, and so on. Because when we solved the ternary structure of that one, we actually found that the MGD, CRBN, or both actually induced a conformational change in the target itself, which now wrapped around the molecule and then also contributed to the PPI interface down here. So what's really interesting about this example is that the conformation that we actually captured in the ternary complex hasn't been observed before. The only conformation that is available on the PDB would be the apostate that is shown up here. And in the apostate, there's only a shallow cavity at best, where later the MGD would then actually carve its own pocket. So I think this example illustrates really nicely how different approaches can actually coexist in this space, and highlights that both rational as well as serendipitous approaches are really have both their justification and should coexist until we have a complete understanding of what's actually possible in this space. And I think what this example also, and also the previous examples that I showed you before, what they highlight pretty nicely is that we have a very exciting and differentiated activities in our compound libraries that now give us all these differentiated binding modes. So that's really exciting for us to see, and we are looking forward to explore this more. Now, to summarize this real quick, I showed you how we are mining the canonical diagram space, and we are predicting over 2,500 targets in that space. We have validated around 120 of these targets. It's not our primary task to validate all the G-loop targets that would be out there, but we have a strong focus on clinically relevant ones, just to mention this here. And I also showed you how we can now use surface mimicry and screening approaches to actually move beyond the canonical diagram space to expand the target space and identify novel opportunities for gluing targets in slightly different ways. But as I also mentioned before, the idea was that to extract basically the learnings from the first two parts and apply them also to move beyond cereblon. And this entire surface analysis is now something that we, of course, cannot only perform for the target side of the ternary complex, but can be also essentially done for E3 ligase substrate receptors. We are not necessarily looking here for surface mimicry, but rather for surface properties that would make these substrate receptors more likely to actually engage in protein-protein interactions. And this is now an analysis that was done for CRBN, where you now see that there's a patch lighting up, which sits right next to the compound binding site. If you do the same analysis in VHL, we don't necessarily detect strong preferences for engaging in PPIs, which could explain some of the differences that we observe in the reprogrammability between the two substrate receptors, at least when it comes to molecular glue degraders. So now this analysis now can be expanded to the entire E3 ligome. And what's interesting is that we are actually identifying substrate receptors and E3 ligase components that outperform even CRBN in their PPI propensities. And we are currently looking into these. We use this analysis to basically to rank E3 ligase components, to select them for basically investigating reprogrammability. And we have a number of these for which we already have nanomolar binders. So now all the science that I talked about before, of course, also feeds into our portfolio. And I hope you can appreciate that it's now a good balance of different binding modes, GSPT1 and NEK7 being G-loop targets, VAV1 and CDK2 representing alternative binding modes here. We have a number of really exciting targets in our discovery pipeline, one of which would be novel binding mode number 5. And we are really looking forward to the day when they finally see the daylight. Great. So that would be it from my side. A big shout out and thank you goes out to the entire team. Of course, it's obviously a huge team effort, and it's always I'm always really impressed how great teamwork can actually, or what great teamwork can actually achieve, where everybody is working like towards the same goal, which is really, you know, coming up with the next generation of pharmaceuticals. So it's always a real pleasure to interact with everybody that you can see on this slide. They are all coming from a different background, and we are all driven by our curiosity and apparently also by the great science. So thank you for listening in, and I'm happy to take questions.